Hello friends, welcome you in Research Needs. In this video, we will see experiments on rat blood sugar. So we will go inside the Ex Pharm software. So now we will click on effect of insulin and Alloxan tab. Now to start experiment, we will click here. Now after clicking to start experiment, we are finding here a well-designed interface. In that we can see here a rat. To collect blood sample, a test tube, a reagent bottle, Alloxan injection, insulin injection, and you can see here glucometer. So this is the arrangement we can find in the interface. So here we can see the objects, principle, experiment, procedure, observation and results. So now we will go and see the objects of the experiment. Demonstration of effect of insulin on the healthy rat, on the rat with insulin dependent diabetes mellitus. So diabetes is produced by administering Alloxan, a substance which destroys the cells from Langerhans isolates of pancreas. So in this experiment we are going to see blood glucose level in healthy rat and then a diabetic rat. Diabetes are going to create here in rats by using Alloxan injection. Now what is Alloxan injection? What Alloxan does? It's a chemical. It destroys the beta cells of Langerhans that are present in pancreas. So this is how we are going to perform the experiment. Now we will see the principle. While we are clicking the principle, we are seeing here two main points that we are going to describe here: insulin and diabetes. Insulin is polypeptide hormones when it is synthesized by the cells from Langerhans isolate of pancreas. The main metabolic action of these hormones is lower blood glucose level by increasing the transfer of glucose across the plasma membranes of the target cell, whereas it enhances glycolysis, inclusion of glucose in glycogen molecules, and transferring of glucose into the lipid and protein. So here it is showing role of insulin into the body. So it is polypeptide hormones that produced from Langerhans isolate of beta cells from pancreas, and its main function is lowering the blood glucose level into the body by use it to produce energy and to in storage of as a glycogen and transfer it to formation of lipid and protein. So here it is showing the insulin functions, its importance in the body. So next we are seeing here what is diabetes mellitus. It is a metabolic disorder which is may be classified type 1, type 2. Type 1 insulin dependent diabetes mellitus. So here we need insulin, and non-insulin dependent diabetes type 2, and which is caused by insufficient synthesis of insulin in pancreas. So here type 1 we are going to produce by giving Alloxan injection and by destroying the beta cell of Langerhans from pancreas. So this type of diabetes we are going to produce in this experiment. And another type is type 2. In this condition insulin is present in the body but it not works, means its receptors are not working, so the glucose is not utilized into the body and its level is increasing in the body. Now here what we are going to do, collect the blood sample from the normal rat and then blood sample from the diabetic rat, and before and after insulin administration we are going to measure the blood glucose. So this was the whole principle of the experiment. Now we will go and see the procedure first. In procedure, we will see, click on the collect of blood sample button, click the add reagent button, click analysis button. The value of the blood glucose is recorded. Click the restart experiment button. Insulin is given by clicking on arrow which indicates the substance, and action from the 1, 2, 3, 4, 5 are repeated. So if insulin injection we have given, before that we have to follow these steps, means 1 to 5 steps again before giving insulin injection. Before giving again here, and Alloxan is given by clicking on arrow which indicates this substance, and action from point 1, 2, 3, 4, 5 are repeated. Means before giving Alloxan injection we have to click these buttons again. So Alloxan and insulin are given by clicking corresponding arrows, and action from point 1, 2, 3, 4, 5 are repeated. So before clicking these two buttons, we have to follow these points. This was the instructions or procedure. Now we will go for experiment. So now we will click on experiment. So it is already, we are in experiment interface. So now we will start the experiment. So to start the experiment, we have to click the collect blood samples. Here we can see the instructions: collect blood sample before and after administering insulin, and then Alloxan, and finally Insulin and Alloxan. So we will collect the blood sample first. So now we will add the reagent. We will click analysis. So here you can see the normal blood glucose level is 86 mg per dial. So now we will go for next step, record the display of glucose level and restart the experiment. So now we will restart the experiment. So it will reset everything. Collect blood sample after administering insulin and then Alloxan. Now we will give injection to rat of insulin. So now we will collect the blood sample. Add reagent. We will take reading in glucometer. It is 76 mg per dial. So here from it is hypoglycemic. Earlier it was normal glucose level 86, and after giving insulin it is reduced 76, means it is hypoglycemic. Now we will go for next step, restart the experiment. We will click Alloxan. We will collect the blood. We will add the reagent. Now we will check the blood glucose level in glucometer. It is 129 after giving Alloxan injection. It has destructed the rat beta cell of Langerhans. So now it, just because of that, blood glucose level is increased. Now we will go for next step, restart the experiment. So first we will give the Alloxan. So we have destroyed rats beta cells of Langerhans. Now blood glucose level is increased. Now we will give the insulin. And we will check the blood sample to check the blood glucose level. So now we will add the reagent and we will do analysis in glucometer. So it is showing 96 mg per dial. So it is reduced near to normal glucose level, but not that much reduced. So it is reduced to 96 mg per dial. That was 129 mg per dial. So now we will see the results. Alloxan increased blood sugar level by destructing beta cells of pancreas, whereas insulin reduces the increased blood glucose level increased due to the Alloxan injection. So this was the result we have seen in this experiment. Now we will see what was be performed during the whole experiment. So here we can compare all the steps. First we have checked healthy rats blood glucose level, that was 86 mg per dial. That status was normoglycemic. Second step was healthy rat with insulin was 76 mg per dial, hypoglycemic. Third stage was healthy rat plus Alloxan 129 mg per dial, hyperglycemic, diabetic. And diabetic rat plus insulin, it is showing 96 mg per dial, means approx normoglycemic. So it has reversed the hyperglycemic condition to normoglycemic condition. So this was all the experiment comparisons. What is the effect of drug, we can easily analyze by this table. So how was the video? Please do comment. Thank you for watching Research Needs. Thank you very much.